Enzymes are found in living organisms throughout nature. They allow for countless reactions to occur within the temperature range that supports life as we know it. Enzymes are biocatalysts, catalysts that are made by living systems or that are themselves living systems. For example, ribulose 1,5-bisphosphate carboxylase oxygenase, or Rubisco, is an enzyme produced by plant cells that catalyzes the first step in the conversion of carbon dioxide gas to glucose during photosynthesis. Alternatively, yeast cells added to bread dough to catalyze the fermentation of sugar are an example of a biocatalyst. It is a living system. Biocatalysis is any method that uses biocatalysts to increase reaction rate. As we learn more about it, people working in an increasingly wide range of fields are becoming interested in biocatalysis. For example, new pharmaceuticals are increasingly complex, which makes them very expensive to synthesize by traditional chemical methods. Researchers are exploring ways to use biocatalysis instead, as a way to reduce costs. Biocatalysis may also play a key role in improving and protecting our environment. Traditional chemical manufacturing processes often produce toxic byproducts. These byproducts are costly, since they must be either stored or converted to non-toxic substances and also have a risk of accidental release into the environment. Biocatalysis has the potential to reduce or eliminate such byproducts. In addition, since biocatalysis can be more efficient than traditional chemical synthesis processes, it may also reduce energy and raw material use. The industrial preparation of paper is an example of a process that may benefit from the application of biocatalysis. The predominant paper pulping method used by the paper industry is the Croft process. Wood is composed of a number of substances. The most abundant and desirable is cellulose, which forms long fibers that give wood its strength. In a living tree, the cellulose fibers are associated with a group of compounds known as lignin. Although important biologically, the presence of lignin lowers the quality of paper. The Croft process removes lignin from cellulose by heating chipped wood in a solution of sodium hydroxide and sodium sulfide. This uses a lot of energy and also produces large volumes of pollutants. Even with restricted emissions, pulp and paper manufacturers that use the Croft process often can be identified by a strong, offensive smell. The Croft process generates hydrogen sulfide gas and other sulfur gas compounds as a byproduct. To produce white paper, paper manufacturers have traditionally used chlorine bleach. However, using chlorine to bleach wood pulp has been shown to produce dioxins, substances that cause cancer in humans and other organisms. Biocatalysts that are proving to be very useful in these applications include the following. Lactases. This class of enzymes can remove lignin from cellulose and wood without the need for harsh chemicals. These enzymes break down the lignin, leaving the cellulose intact. Xylanase. Treatment with this enzyme enables manufacturers to reduce chlorine consumption by 15–25%. This enzyme is used in bleaching procedures in which chlorine is completely replaced with chlorine dioxide or non-chlorine chemicals. The paper and pulp industry has had a very significant impact on the environment, which has affected many decisions in the industry as well as behaviors at personal and business levels. Due to technological advances like the printing press and the harvesting of wood, there has been a high availability of disposable paper, which has also led to high consumption and waste levels. Due to its affordability, examples of the environmental impacts of the pulp and paper industry include air pollution. The pulp and paper industries produce nitrogen oxides, sulfur oxides, and carbon dioxide, in which nitrogen and sulfur dioxides contribute to acid rain and carbon dioxide contributes to climate change. Water pollution. Pulp and paper mills are sources of nutrients, solids and dissolved matter like lignin in wastewater discharges. There are other agents like alcohol or inorganic materials such as chlorates. These pollute the rivers and lakes that they are released in. Paper waste. About 26% of solid waste dumped in dumping sites is discarded paper and paperboard, which take up a significant amount of space in landfill sites, causing more pollution. Also, when paper is disposed of in dumping sites, it subsequently breaks down, and methane is produced, which contributes to global warming. Chlorine and chlorine-based materials. In bleaching wood pulp, chlorine and its compounds are used. However, the use of chlorine and its compounds produces a lot of dioxins, which are persistent and highly toxic pollutants. 40% of the world's commercially cut timber is used for the production of paper. Pulpwood plantations and mills endanger natural habitats. Over 30 million acres of forest are destroyed annually. The pulp and paper industry is a big contributor to the problem of deforestation and is partly to blame for the endangerment of some species that live in the forests. Paper production uses up lots of water, and A4 paper requires 10 liters of water per sheet. The paper industry is the 5th largest consumer of energy in the world. It uses up 4% of the world's energy. To produce 1 ton of virgin paper, it is estimated that 253 gallons of petrol is used. In the pulp and paper industry, the most important application of enzymes are: present-day bleaching of kraft pulp uses large amounts of chlorine and chlorine chemicals whose byproducts are toxic, mutagenic, persistent, and bioaccumulating and cause numerous harmful disturbances in biological systems. Enzymes provide a very simple and cost-effective way to reduce the use of chlorine, chlorine compounds, and other bleaching chemicals. For example, in the pre-bleaching of kraft pulp, xylanase enzymes have been found to be most effective for this purpose. So far, 2 methods have shown promise in the use of enzymes for bleaching, one using hemicellulase enzymes, and the other ligninolytic enzymes. Hemicellulase enzymes are used commercially in pulp bleaching. The main enzyme needed to enhance the delignification of kraft pulp is reported to be Endo-Greek letter beta xylanase, but enrichment of xylanase with other hemicellulosic enzymes has been shown to improve the effect of enzymatic treatment. However, ligninolytic enzymes, unlike xylanases, attack lignin directly, and hence are more effective. Enzymatic modification of fibers aims to decrease energy consumption in the production of thermomechanical pulps and increase beatability of chemical pulps and improvement of fiber properties. In high-yield mechanical pulps, most of the lignin and hemicellulose remains in the pulps. Xylanases are applied to thermomechanical pulp, and when combined with an alkaline pretreatment, the amount of energy required for refining the thermomechanical pulp was decreased. Enzymatic deinking is a new approach to convert secondary fibers into quality products. It has been proven to be an effective and economical method of deinking waste paper. Drainage enhancement is also another benefit of enzymatic deinking. Moreover, the enzymatically deinked pulps possess superior physical properties, higher brightness, and lower residual ink compared to chemically deinked recycled pulps. Also, the size distribution and shape of ink is controlled using the enzymatic process to increase the efficiency of the size-based flotation process. This is done by selectively varying enzyme composition, charge, and residence time, as well as varying other additives and pH in the system to effectively dislodge the normally large, flat, and rigid ink particles into much fewer and non-platelet forms. The enzymatic deinking process also improves freeness compared to chemically deinked pulps. Pitch is a mixture of fatty acids, resin acids, sterols, glycerol esters of fatty acids, other fats, and wax is usually found in wood and is soluble in methylene. It is less than 10% of the total weight of wood but causes major problems. Pitch reduction with enzymes is a very efficient biotechnological method. Different lipases have been used for removal of pitch. Enzymatic pitch control helps to removes pitch-related problems such as reducing defects on paper web as well as the frequency of cleaning pitch deposits in the paper machine. It also has other advantages such as using eco-friendly and non-toxic technology, improved pulp and paper quality, reduction in bleaching chemical consumption, reduction of effluent load, and space and cost saving in a millwood yard by using unseasoned logs. Enzymes have also been used to increase pulp fibrillation and water retention and to reduce beating time in virgin pulps. With recycled fibers, enzymes have been used to restore bonding and increase freeness. Specialized applications include the reduction of vessel picking in tropical hardwood pulps and the selective removal of xylan from dissolving pulp. Enzymes have also been investigated for removal of bark, chives, pitch, and slime and for retting of flax fibers. According to Pulp and Paper Canada, the pulp and paper industry is currently focusing on researching enzymes that can modify wood into biopolymers and materials of higher value, such as increasing the reactivity of cellulose, hemicellulose, and lignin. The industry is trying to identify the strands of DNA that contain the code for desired properties in enzymes, such as higher stability, using phylogenetic analysis and sequence similarity networks to analyze DNA. The DNA is then grafted into host organisms such as fungi or bacteria to mass-produce improved enzymes. The ultimate goal of the research conducted is to create a biocatalyst toolkit that will record the genetic information, enzyme activity, and operation conditions discovered and store it in the Characterized Lignocellulosic Active Enzymes Database, CLE. By using enzymes, the industry is able to reduce its AOX, high levels of which have been related to pollution, discharge by 25%, which is 2,700 tons of AOX annually. With the introduction to the mechanical pulping industry, there is the potential to save 632 GWH yearly. By incorporating biocatalysts into the mechanical pulping industry, it is also estimated that there will be 155,000 to 270,000 less tons of CO2 emitted. Chemical catalysts can accelerate delignification, and they show promise as a new technology that can achieve better selectivity and fiber yield at equal or lower cost than conventional methods. Cobalt-based oxidative catalysts are attractive leads because they, 1. Operate under mild conditions, 2. Use plentiful O2 and non-precious transition metals, and 3. Produce valuable chemical feedstocks as byproducts. Different aspects of economic considerations are among the most dominant topics related to assessing the feasibility of process substitution towards enzymes. These range from strict cost-effectiveness considerations, via issues related to the risk involved in trial processes, problems with estimating future benefits, the need for investment in new production equipment, and to international market concerns. The first and most important factor is the production cost. Clean technologies can be cost-saving due to the reduced consumption of resources, reduced costs for safety, cleaning and remediation, etc. These are all factors that have a direct impact on the bottom line of a company. But if the enzymes themselves are significantly more expensive than the traditional chemical alternative, the indirect savings rarely equal the added expense. Kraft pulping is energy-intensive and it provides a less efficient use of wood resources than desired. Catalytic methods offer a viable alternative to increase cellulose fiber yield by improving selectivity, reducing energy use and eliminating the formation of odor-causing mercaptans associated with kraft pulping. If successful, catalytic methods will improve yield, reduce fiber waste, reduce energy use, lower the cost of bleaching and decrease the cost of air emission controls. The purpose of this CRADA was to combine industrial expertise in wood pulping with national laboratory expertise in high-performance computing, eventually leading to the design of new catalysts that can remove lignin from wood pulp efficiently and cost-effectively. Pitch is composed of fatty acids, resin acids, sterols, glycerol esters of fatty acids, other fats, and waxes, and is usually defined empirically as the wood component that is soluble in methylene. It is less than 10% of the total weight of wood but causes major problems. Pitch reduction with enzymes is a very efficient biotechnological method. Different lipases have been used for removal of pitch. Enzymatic pitch control helps to reduce pitch-related problems to a satisfactory level. It reduces defects on paper web as well as the frequency of cleaning pitch deposits in the paper machine. At the same time, it also offers other advantages, such as eco-friendly and non-toxic technology, improved pulp and paper quality, reduction in bleaching chemical consumption, reduction of effluent load, and space and cost saving in a millwood yard by using unseasoned logs. Present-day bleaching of kraft pulp uses large amounts of chlorine and chlorine chemicals. Byproducts from using these chemicals are chlorinated organic substances, some of which are toxic, mutagenic, persistent, and bioaccumulating and cause numerous harmful disturbances in biological systems. Enzymes provide a very simple and cost-effective way to reduce the use of chlorine, chlorine compounds, and other bleaching chemicals. Enzymes also offer a simple approach that allows for a higher brightness ceiling to be reached. So far, 2 enzyme-based approaches have been investigated. One uses hemicellulase enzymes, and the other uses ligninolytic enzymes. Enzymes are being used in the pulp and paper industry to reduce energy and chemicals by breaking down or degrading the polymeric components of woody biomass into smaller, simpler molecules. Due to the ease and efficiency with which enzymes can be used in the industry, the global market for enzymes in the pulp and paper industry has been growing to over $250 million per year. Another reason why there has been an increase in the use of enzymes is the increased availability of a whole range of enzymes at reasonable cost. To name a few, the enzyme xylanase has seen an increase in the industry. Today, about 10% of all kraft pulp is manufactured with xylanase pre-bleaching. In North America, Iogen Corp., based in Ottawa, has established a market leadership position in the use and distribution of xylanase. Globally, other suppliers such as AB Enzymes, and more recently Diversa, are also selling to this market. In Japan, Oji Paper is unique in manufacturing xylanase on-site at its Yanaga mill. Biopulping has also seen an increase in the use of enzymes in the industry such as syllobia hydrolase. This enzyme is available in fairly pure form from AB Enzyme thanks to a cloning program at the VTT, Helsinki labs. A full-scale trial was run at the UPM Kemienrauma mill in Finland and a savings in energy was reported during the trial and in associated pilot work. In conclusion, with the continued increase in the awareness of the efficiency and reliability of the use of enzymes in the pulp and paper industry, there is likely to be a drastic increase in the use of enzymes to further advance the paper and pulp industry in the future. Xylanase and lacase are enzymes, which are proteins. Proteins can only function within a small pH range. Otherwise, they can become denatured. When an enzyme becomes denatured, it can no longer perform its task, as the structure of an enzyme is directly related to its function. Also, in addition to pH, proteins can become denatured because of extreme temperatures. Wood pulp produced by the kraft process is typically highly alkaline, and may reduce certain types of xylanase and lacase useless. This means that extensive research must be done by companies in order to decide upon a specific type of biocatalyst that can function under the conditions of the mill, or alterations must be made to the process itself. Both options can be timely and expensive for companies. Another potential drawback of biocatalysts is the possibility of enzymes degrading the quality of the pulp. Xylanase, for example, reduced the tensile and burst strength of the pulp produced at the Marathon mill in the aforementioned study by 8% and 6% respectively. Thanks for watching. This video is a part of UofT's class of 2025.